All right, we're going to talk about the nature of genes. Basically, when we talk about the nature of genes in molecular biology, information always flows in one direction. So, we call this the central dogma. Genes is the genetic material that's going to be in chromosomes in our nucleus. So the central dogma of molecular biology states the information flows in one direction. First of all, you have to take your DNA and you have to make a copy of it. So DNA is going to be copied. Really, it's going to be copied into mRNA, messenger RNA. And your mRNA is going to leave the nucleus and it's going to travel to the ribosome where it's going to be read and it's going to be flowing into a protein. So when we talk about DNA being duplicated, that would be replication. So we're going to use new terms. When you use the word transcription, the word transcription literally means the flow of information is going to go from DNA to RNA. When you use the word translation, you're going to take your RNA and you're going to read your RNA and it's going to create a protein. So replication's going from DNA to DNA. Transcription, on the other hand, is going from DNA to RNA. Now, we are going to talk about the 3 different types of RNA. When we talk about transcription and translation, when we look at a cell, your nucleus is going to be responsible for doing your transcription. So, when you take your DNA and you make it into mRNA, that is a process of transcription. That's going to occur within the nucleus. So, in the nucleus is the site of transcription because your DNA can't leave. So originally, whenever we talk about mRNA is a copy of DNA. So transcription occurs inside of the nucleus. Now, your mRNA will leave and it'll go to a ribosome. The ribosome is responsible for protein synthesis. So, your mRNA, the copy of your DNA, exits and it attaches to your ribosome. And your ribosome is going to read your mRNA. And what it's going to do is it's going to produce a protein. So as it reads your mRNA, amino acids are going to be linked together to build a protein. When you build this protein, when you read mRNA, when you take the mRNA and you convert it into a protein, That is called translation. Now, the site of translation is really going to be at the ribosome, considering it's going to make the protein. If we were going to practice looking at this, let's just say in our nucleus, our DNA strand reads GCA, TAC, GAT, TAC. If we were going to make mRNA, we would make a copy of DNA. It has to be complementary. So, during the process of transcription, you're going to make mRNA. It has to be complementary to the DNA. We always read these in sets of 3, and that'll come up here in a moment. So, in RNA, our adenine is always going to pair with uracil, and our guanine always is going to bind with cytosine. So, one key difference between RNA and DNA is DNA has thymine, mRNA doesn't. But they are going to be complementary base pair. So, first one, when you transcribe your DNA, it would be CG. Now, A complementary binds with U. So anytime you have an A, you're going to put a U on the opposite side. But since you don't have a T, T would be binding with A anyway. You have A's. So you place that with A, U, G. You go to the next 3. C, A binds with U. T is not in RNA, but T normally binds with A in DNA, so that would be an A. And then A, U, G. Put that twice. That's transcription. Now, the ribosome is going to read mRNA to figure out which proteins you need. Now, proteins are made of amino acids. So in our book, we have a something that basically helps you read mRNA. You will never have to basically read mRNA, but you use what we call the genetic code. So during translation, the ribosome is going to translate. So basically it reads mRNA and it translates the amino acids that we need to use to build a protein. So, during translation, the ribosome reads CGU. It is going to build a protein by saying, oh, okay, I need the amino acid, whatever it is. It's reading it. We're going to go over the genetic code here in a minute. Now, what it does is it takes the amino acid and it is going to put a bond called a peptide bond in between the amino acid so that it can read the next 3 RNAs. AUG encodes for amino acid I know, which is methionine. So the ribosome again is reading your mRNA and determining which amino acid is going to be needed to build this protein. So a protein is amino acids bound together by peptide bonds. So then it builds another peptide bond. The ribosome reads the next mRNA, and it's going to place another amino acid. It builds another peptide bond, and we build another amino acid. So a protein, when we talk about the word protein, a protein is basically also called a polypeptide. A polypeptide is amino acids bound together by peptide bonds. So that is how we build proteins. Amino acids are bound together by peptide bonds, and in translation, we say that translation is taking mRNA to build a protein. So it all goes back to the words. Transcription is going from DNA to RNA, and translation is reading your mRNA at the ribosome and converting it into a protein. And the way we convert it into a protein is we take amino acids and we put peptide bonds in between it, and we build a protein. So that is the central dogma in the flow of genes. Now I want to talk about the genetic code.